In this video, I'm going to give you an overview and an introduction to hormones. Well, let's begin with the basics. What are hormones? Well, hormones are a diverse group of molecules. They're typically produced in specialized cells in endocrine tissues, but not always. They're carried in the bloodstream and they act on distant tissues. They're one of two types of signals that the body uses to coordinate physiological responses, the other one being the neurotransmitters of the nervous system. In general, the nervous system is used for quick and really specific communication between two sets of cells, whereas hormones are best suited for slower acting, widespread coordination of activity. Hormones play a key role in a whole bunch of processes, including growth, development, reproduction, metabolism, and by that I mean not only food and energy balance, but also temperature balance, and water, ion, and electrolyte regulation. Well, earlier I said that many hormones are produced by specialized endocrine tissues. Here's a cartoon that shows us some of these. Now, some endocrine tissues are found in the head. The pineal gland is in the brain itself. The pituitary gland is closely associated with the brain, but other endocrine tissues are scattered through the body. Let me also add that in addition to the structures shown in this cartoon, there are other structures not really considered endocrine structures that also release hormones. So for example, your fat cells release a compound called leptin, which acts as a hormone. And there are a bunch of different hormones that are synthesized and secreted by various parts of the digestive system. There are a lot of different hormones, but we can break them into three broad classes: peptide hormones, steroid hormones, and then what I'll call single amino acid derivatives. Let's look at them one by one, beginning with peptide hormones. Well, as the name suggests, peptides are small, water-soluble proteins. They can be as short as 3 amino acids in length or as long as about 200. They're usually encoded by a single gene, but there are some hormones that are dipeptides made up of 2 different subunits, each encoded by a different gene. Like all proteins, they're synthesized on ribosomes, and then because they're going to be secreted from the cell, they pass through the ER to the Golgi to secretory vesicles, and then they'll be released upon stimulation. Here's a little cartoon. I'll walk you through this. So remember, the genes are in the nucleus, and transcription will begin in the cytoplasm, but then secreted proteins move from the ER to the Golgi, and then are stored in these secretory vesicles until they can be released. Along the way, a number of things can happen. So for example, hormones are often synthesized as longer prohormones that are then cleaved to produce active hormone. Secreted proteins can be glycosylated, having sugar chains added to them in the ER and Golgi, and sometimes this happens to hormones, and for other hormones it doesn't. And then finally, some hormones are packaged along with carrier proteins so that when they get released, there's also a compound that will help move them through the bloodstream. Regardless of all of this, when we think about peptide hormones, we think about them as being hydrophilic, polar molecules, and they're going to act by binding to cell surface receptors. Because this group is pretty diverse, let me give you some examples of the types of peptide hormones that we find, because we can divide them into several subclasses. The glycoproteins, that includes follicle-stimulating hormone, FSH, thyroid-stimulating hormone, also called thyrotropin, luteinizing hormone, LH, and human chorion gonadotropin, HCG, are a group of heavily glycosylated dimers, and here they are made up of 2 subunits. So there's a green subunit and a red subunit, and they're glycosylated, and that's what I'm showing with these yellow dots, the sugars that have been added as part of the processing of this protein. The POMC family provides a nice illustration of prohormones. Everybody in this group is derived from a single protein product that gets cleaved into multiple active peptides. Here's our prohormone here, it's called POMC, and then that can get clipped to yield beta-lipotropin and another prohormone, which can be clipped again to yield ACTH, adrenocorticotropic hormone. That's an important component of the pathway that's going to release stress hormones like cortisol. It can also be clipped to release alpha-melanostimulating hormone, which is involved in feeding and pigmentation and sexual behavior. Other products include beta-endorphin, which plays a role in pain, but also in other physiological functions. A third group would be the insulin and insulin-like growth factor family. And this is another example of how a hormone can be processed, because although in the final product insulin has 2 different subunits, shown here in green and blue, This is initially synthesized as a single protein that then gets clipped to yield these two subunits that are held together by the disulfide bridges. More peptide hormones include the growth hormone family. That includes growth hormone and also prolactin. These are the largest of the peptide hormones. They're about 200 amino acids in length. They're unglycosylated, but they have a lot of tertiary structure. They're held together by internal disulfide bonds. And last but not least, there's the posterior pituitary hormone group, oxytocin, and antidiuretic hormone, ADH, which is also called vasopressin. These are nonapeptides, meaning that they're only 9 amino acids long, and they're very similar. They differ only in 2 amino acid sites. All right, let's move on to the steroid class of hormones. Steroids are synthesized from cholesterol, and they are going to be nonpolar. So this sort of scary figure here shows us the synthesis pathway for steroid hormones, beginning with our starting material, cholesterol. And what each one of the arrows represents then is an enzyme that can go and modify that. All of the steroid hormones share a 4-carbon-ringed core, but they're going to differ in the side groups and the covalent bonds. That's what the enzymes will be acting to modify. The particular steroid hormone that gets produced in a cell will depend on which of these enzymes is active. So for example, if we have a cell and the 2 enzymes that are catalyzing the 2 steps that I've highlighted in black arrows are present, then we'll be able to produce progesterone. A different cell might have those 2 hormones as well as 3 more and then could produce testosterone, and yet another could then have an additional enzyme aromatase and be able to produce estrogen. These enzymes are important for another reason, and that's because all of these steroid hormones are lipophilic. They're nonpolar and they're able to pass freely out of the membrane. So they will be synthesized only when the endocrine cell is stimulated, and the way that that stimulation will then lead to the production of steroid hormones will be by activating these enzymes. And let me just repeat that they're going to bind primarily to intracellular receptors because they're nonpolar molecules. Our last class of hormones are the single amino acid derivatives. Now most of these, epinephrine, norepinephrine, dopamine, and serotonin, they're produced from a single amino acid precursor through an enzyme-catalyzed reaction. Let me give you an example here. Here's our starting material, tyrosine, an amino acid. And if these 2 enzymes are present, then we can produce dopamine. If a different cell has those 2 enzymes plus another one, it can produce norepinephrine. And if all 4 of these enzymes are present, it'll be producing epinephrine. All of these compounds that are produced are polar. They're going to act by binding to cell surface receptors. The other hormone that gets included in this class of hormones is thyroid hormone. Now, thyroid hormone is really pretty different from the other amino acid derivatives. Thyroid hormones are produced when 2 tyrosines in a precursor molecules are iodinated, enzymatically fused, and then clipped from a precursor molecule. Now that's a mouthful, so I'm going to try and explain it to you using this relatively complicated cartoon. Thyroid hormone is produced in the thyroid gland, and so thyroid cells here are shown in purple, and what I want you to notice is that those cells are producing a protein called thyroglobulin. The thyroglobulin protein contains a number of tyrosine residues. It's one of the amino acids. The cell does something kind of weird. It takes this thyroid hormone and it secretes it into what's called the follicular space. And there, the tyrosine residues get decorated with iodine, and there's also something called conjugation. And what happens there is that one of the tyrosines is clipped off and then covalently linked to another tyrosine in the same protein. Once that's done, the cell takes the protein back up inside using endocytosis, and there it gets processed further. What happens is that the 2 iodinated tyrosine molecules, the thyroid hormones themselves, get clipped away from the rest of the protein. The bulk of the protein is completely discarded. It gets broken down, and the thyroid hormone then is released into the bloodstream. Thyroid hormone is unusual in another way. Even though it has a carrier protein in order to help it pass through the membranes, it's going to bind to intracellular receptors. Okay, well, I just mentioned receptors, so let's look at those in a little bit of detail too. Most hydrophilic hormones bind to what are called G protein-coupled receptors, GPCRs. You may have seen these before. GPCRs are integral membrane proteins. They have 7 membrane-spanning domains, and they change configuration when a ligand binds. They're associated with heterotrimeric G proteins, and that can initiate intracellular signaling cascades. So this is how they're going to work. We'll have our receptor, and in the inactive state, it's associated with the heterotrimeric G protein with the alpha, the beta, and the gamma subunits. In the inactive state, the alpha subunit is associated with GDP. Now, if we add a ligand, in this case the hormone, what will happen is it binds to the receptor, there's a conformational change, and when this happens, the alpha subunit releases the GDP and binds a GTP instead. As soon as that happens, the heterotrimeric G protein dissociates, so the beta and the gamma subunits go floating off and the alpha subunit goes floating off, where they can interact then with effector molecules, and often these effector molecules initiate enzyme cascades, leading to the activation of some suite of proteins in the cell. This is the most common type of hormone receptor. Many different hormones act through GPCRs. The hormones that do this include the adrenergic receptors, so epinephrine, norepinephrine, the glycoprotein hormones, thyroid stimulating hormone, luteinizing hormone, follicle stimulating hormone, glucagon receptors, muscarinic acetylcholine receptors, so not all acetylcholine receptors, but the muscarinic class, parathyroid hormone receptors, and most serotonin receptors are GPCRs. And you might ask yourself, how can so many different kinds of hormones act through the same system? And the answer is that there is a lot of variation in it. So there are a lot of different GPCRs. Each one of these hormones has a unique GPCR receptor. And in addition, the GPCRs can be linked to different cell signaling pathways by activating different G-alpha subunits. So some hormones will activate the G-alpha S or the G-alpha I class of G proteins that can either increase or decrease the activity of an enzyme called the adenylate cyclase, leading to the production or an activation of protein kinase A. Other G proteins will act through what's called the G-alpha Q11 class, and this leads to a release of calcium into the intracellular space and activation of a different protein kinase. Still other G proteins act through the G-alpha 12-13 class. And sometimes the beta and the gamma subunits can go off and have their own action. So different kinds of hormones can turn on different effects in different cells depending on what the GPCRs are coupled to. You should also know that there's a lot of variation in what the GPCRs themselves look like. Here I'm showing you a picture of the GPCR that acts as a receptor for follicle stimulating hormone, FSH. And what I want you to notice over on the left side of the figure is that the extracellular domain of this compound is huge, and that's the part that will be associated then with binding follicle stimulating hormone, ultimately leading to the activation of the GS, adenylate cyclase, cyclic AMP protein kinase A pathway. Other hydrophilic proteins act through what are called the insulin or IGF receptors or receptor tyrosine kinases. Here what I'm showing you is an insulin receptor, and this class of receptor will change conformation when the hormone binds. So here it is in an inactive state, I throw on insulin, and the cytoplasmic domain changes shape. Now these receptors have an intrinsic tyrosine kinase activity, which means that they can go and add phosphates to tyrosine residues, and as a result, the protein receptor itself can be phosphorylated. And phosphorylation then can lead to the recruitment of adapter proteins, which can then initiate cell signaling cascades. So I'll show you this for insulin here on this cartoon. We've got the insulin receptor up at the front, and you can see that its activation has allowed it to recruit either this SHC or the IRS adapter proteins, and those two in turn can then turn on a suite of intracellular messengers that can lead to cell growth, changes in gene expression, or changes in metabolic pathways. Hormones that act through this type of receptors or receptors that are related to them include insulin, then insulin-like growth factor, atrial natriuretic peptide, which is involved in regulating blood pressure, and then activin and inhibin, which are involved in regulating the activity of FSH and other hormones in the pituitary. The last class of hormone receptors I want to talk about will be intracellular receptors. This class of receptors will include the receptors for all steroid hormones. All steroid hormones can diffuse freely through the membrane and bind to intracellular receptors. So out here we have a steroid hormone, it might be estrogen or testosterone, and it can pass through the membrane into the cytoplasm. In the cytoplasm we'll find the hormone receptor, and that's often associated with the chaperone protein, in this case a heat shock protein. And when the hormone enters the cell, it can displace the chaperone protein and then bind to the hormone receptor. And this complex then can move to the nucleus and act as a transcription factor. It sits down on the DNA, it binds to what are called hormone response elements, and it can turn on or off transcription of specific sets of genes. And this process often involves co-regulators, so other factors that are involved in this transcription factor complex. Thyroid hormone also has an intracellular receptor, but it acts a little bit differently. Thyroid hormone is transported into the cell, and the thyroid hormone receptor itself is nuclear. It's sitting in the nucleus. But in the absence of the hormone, the thyroid hormone receptor is bound to co-repressors, and so there's no gene activation. And when the thyroid hormone then enters the nucleus and can bind to its receptor, there's a conformational change and gene transcription can begin. You might notice that in this cartoon, this pathway is labeled the genomic pathway, and the reason for that is because both thyroid hormone and steroid hormones sometimes can act through non-nuclear receptors. So depending on the hormone, these might be found in the cell surface or in the cytoplasm. In this case, thyroid hormone can bind to a thyroid hormone receptor in the cytoplasm and affect the activity of protein kinase G. What hormones act through this class of receptors? Well, thyroid hormone and all the steroid hormones, estrogen, testosterone, progesterone, cortisol, aldosterone. But in addition to these hormones, this is also how vitamin D3 and retinoic acid work, and how fatty acids and bile acids can also affect metabolism. This has been a very quick look at hormones. If you continue with this, then you'll want to go and study individual hormones to learn all the details of their actions. As you do that, I encourage you to think about what cells are producing it. So where's it coming from? What's the structure of the hormone? And then what's the structure of the receptor? Because taken together, those will give you some insight into how those individual hormones act.